Good afternoon, everyone. My name is Esther Cho, and I'm a student in the Department of Molecular Genetics at U of T. And today I'll be sharing with you all a novel virus that we've discovered and how this opens up our understanding of viral residency, as I like to call it. So, Dulcivirus diffusus was first detected in RNA-seq data from HPV-positive HeLa cells. Now, HeLa cells are a human cervical cancer cell line, and it can be used to study how HPV causes the majority of cervical cancers. Differential expression upon HPV infection can reveal novel therapeutic targets, which is one major motivation for the study. Now, our virus was detected because this RNA-seq data had a novel palm signature. But why? Why would a non-HPV signature be detected in HPV-positive cells? To skip ahead a little bit, D. diffusus is a kind of narnavirus. Narnaviruses are single-stranded RNA viruses with a singular genome that primarily infect fungi, but also plants, insects, and protists. And what's really fascinating about this virus is that although there are only two formally recognized species, both in budding yeast, hundreds of other species have been sequenced and characterized. And it turns out that this signature maps to 9 other bioprojects and 12 datasets. And looking at the associated species, wild animals in China, flowers, fruits, and bees, it's puzzling how D. diffusus could be picked up so diversely. Given this, and knowing that these species don't transmit narnaviruses across one another, we hypothesize that D. diffusus infects plant-associated fungi and resides in various organisms via environmental deposition and not infection. To study this, we did initial correlation studies to see what other viruses are picked up by searching the SRAs in Serratus and RDRP signature on BLASTP. Now, assembling the genome was challenging because all reads had extremely low coverage of around 1 to 10, and the contigs were heavily fragmented as well. But we found one method that worked best, and it was assembling longer contigs before blasting the palm sequence to them. And to do this, we used MegaHit, which is a software where we could assemble larger contigs, and Diamond to align shorter contigs with higher sensitivity. And compiling the two together extracted the most amount of sequence. Next, to see what host D. diffusus may have, we looked at its phylogeny, where we did multiple sequence alignments of the aligned RDRPs from Protein Blast and visualized the resulting tree. So first, in our initial correlation studies, we found that the RDRP signature aligned most significantly to fungal narnaviruses, some of them being plant-infecting fungi, which is consistent with our hypothesis. Next, our recovered D. diffusus sequence, where we used MegaHit and Diamond, suggested that it has a single ORF encoding the RDRP and no other protein domains, which is consistent for narnaviruses, and we also found the canonical GRR motif within its palm region. But as you can see here by the blue line, we suggest that this did not quite recover the entire genome, or even its entire RDRP. So, upon blasting its recovered protein sequence, we found that all 83 alignments with other viruses happened at their C-terminal ends. This was pretty consistent with how we didn't see motifs A or B upstream, and how the RDRP was half the length of a typical narnavirus RDRP. While divergence could be an explanation, the low coverage makes us believe it's a detection issue. And because of this, we couldn't interpret our phylogenetic tree with high confidence. So going back to BLAST to see what we could see with their recovered RDRP, we found that our initial list now includes alignments with more protists or plant-associated narnaviruses, and not strictly fungi or plant-associated fungi. So our findings overall open up the possibility of D. diffusus infecting plants and not fungi as hypothesized, or perhaps a novel way in which both can be infected, but it also shows largely how viruses can be detected across species without direct transmission or sample contamination with the residence theory that I suggested in the beginning. And much is still left to be discovered. Could the N-terminal region actually have the AMD motifs? Could this virus actually integrate in more hosts rather than residing? And of course, how can we apply these methods to study more viruses? Over time, thousands of novel viruses have been sequenced and characterized, but there's still a lack of understanding of how different narnaviruses can be transmitted or reside based on its species. Ultimately, the work towards discerning between direct, indirect, and passerby viruses and how they reside across various organisms with regards to infection and pathogenesis is a very exciting avenue to keep exploring.